Okay. Well, let's go ahead and get rolling for today. So, in the way of announcements, I posted your problem set as I have said I would do. I posted that on Canvas, so make sure you print that off. As of right now, we'll have that be due on the 8th. I think that we'll be through the material by Wednesday well enough so that you could do that, okay, that problem set. If we get slowed down or bogged down with material, then we'll have to delay that to, unfortunately, the same day that the exam is due. So I don't really want to do that, but that might be the case. Okay. Second thing is that we'll have our group work on Monday. So we're going to hopefully start glycogen metabolism today a little bit. I'll kind of introduce you to the structure of glycogen. And then on Monday, you'll come in and do a worksheet on glycogen breakdown and synthesis. So come prepared for that. That group work is the one that I know historically has taken the groups longer to get through than certainly the first two. Okay, all right. Our riddle here, let's go ahead and jump into that. This one's a difficult one. 500 begins it, 500 ends it. 5 in the middle is seen. First of all figures, the first of all letters, take up their stations between, join all together, and then you will bring before you the name of a king. What is the name of that king? David, exactly right. Exactly right. David. And you all say, where do you get David from that? So the Roman numeral letter, or the Roman numeral for 500 is D. Okay, so we have D and D, and then there's 5 in the middle, so that's Roman numeral V. And it says first of all letters, of course, is A, and then first of all figures is the Roman numeral 1, I. Some of you are shaking your head like, no, I just stop. Okay, anyway, I'm not going to stop. I'm going to keep doing it. Okay, all right, now let's go ahead and go here. We are right here. Yes, so this is where we are at. Okay, so I'm going to back up to this slide right here. So on Wednesday, we essentially finished a discussion on looking at how changes in particular in glycolysis and how regulation of glycolysis can be done in a cell in which the energy demand can change drastically within seconds of timescales, right? So skeletal muscle, if you go from resting to a sprint, can have this huge increase in this need and demand for ATP. And because of that, then the muscle cell has to be equipped to increase metabolic rate through, in particular, glycolysis, which is the fastest way to produce ATP. It has to be able to do that in very short periods of time and also do that in a fairly major way. Okay, and we looked at the fact that glycolytic rate can increase actually 90 to 100-fold in these cells within seconds of timescales. Okay, and the breakdown products of ATP as well as ATP itself are some of the most important regulators of this pathway within this cell type or this tissue type. AMP is very important. It will activate PFK1, which will then produce more fructose 1,6-bisphosphate, which will feed forward and produce activate pyruvate kinase, and that will increase flux through this pathway. Okay. So that would be one aspect of this. The other aspect of this that we talked about on Wednesday was the substrate cycle, the fact that FBPase1 is present in these cells. And at rest, FBPase1 essentially takes any fructose 1,6-bisphosphate, not all of it, but most of it, and just simply recycles it back to fructose 6-phosphate. And so there's this kind of this recycling of that. Those carbons, they don't really go through the glycolytic pathway. There is certainly some that leak through, but flux through the pathway is really, really low at rest. It's like I said, it's about 1-ish, okay, if we were to use the numbers that I used on Wednesday, okay. Compare that to when AMP concentrations go up, that flux through the pathway, because FBPase1 becomes inhibited and PFK1 becomes activated, can go up like 89 to 90-fold. Okay, so pretty remarkable actually when we start analyzing it that way. Now, interestingly, I wanted to go ahead and address this. I talked to my TAs yesterday, and one of the TAs had the interesting question, well, why not, instead of having just FBPase1, why not just super reduce the activity of PFK1 to really low values instead of having to continually recycle it? And so I think the best answer for that question is likely due to the fact that it might be impossible to decrease the rate of PFK1 to that level, like 1% of its activity. Based off of the levels of ATP in the cell and also just physical limitations of PFK1. So that's probably the best answer for that, if you were wondering, if you had that same question. Okay. All right, so what we want to do today, let's go ahead and jump forward, and we're going to do another one of these things that I call special topics, and this one is going to keep coming up in the class. We're going to look at diabetes, diabetes mellitus. We're going to look at this for a number of reasons, and this will come up time and again throughout the semester whenever we really get to new metabolic pathways, because diabetes is a major metabolic abnormality, and certainly a disease that has become more and more prevalent and more important from a health standpoint, at least in Western societies. Okay, the incidence of diabetes has gone up significantly over the last couple of decades. So, well-studied disease state. Certainly a common disease state within Western societies. Not only that, but it's also a really great way to look at metabolism and try and understand metabolism at a deeper level, because we can look at these abnormalities and even understand the normal metabolism. Okay, so that's what we're doing here, and that's what will keep coming up in terms of diabetes. I think I would venture to say that all of you have some concept of what diabetes is, but real quickly we'll just give kind of a clinical definition of diabetes. Diabetes would be characterized by a fasting glucose level of about 7 millimolar or higher. That would be really quite high in a fasted state of the individual. Or, a little bit more objective would be doing a glucose tolerance test. Administer 75 grams of glucose to the patient, take their blood glucose 2 hours later. If they have levels of glucose above 11 millimolar in their bloodstream, then they're going to be diagnosed with diabetes at that point in time. Okay, so the characterization of this disease would be higher than normal blood glucose, and really the inability of the individual to clear glucose from the blood. Okay. That can lead over a long, sustained period of time to really chronic pathologies that would be quite horrible. It can lead to significant kidney damage, damage to the retina, damage to the extremities, okay, to the point where these individuals have to have things amputated. They can often go blind. They might have to be on dialysis for much of their life. So really a terrible disease from that standpoint. In an acute sense, it will result in these two things. I think it's glucoseuria. I'm not sure I'm pronouncing that exactly right, but I do know that it means that there would be glucose monitored or found in the urine. All right, so of course, high blood glucose levels at for a sustained period of time, that's going to be filtered through the kidneys and into the urine. Okay, and so you would monitor or see glucose in the urine, which is not normal. Ketoacidosis would be something else. We'll talk about the basis of ketoacidosis later on in this class when we get to ketone bodies in our discussion on lipid metabolism. So the liver, a healthy liver, will produce ketone bodies at a certain level within any individual. In untreated diabetes, it will produce far too much ketone bodies, and that can lead to actually changes in blood pH, which can actually be very dangerous. Okay, so ketoacidosis would be acute pathology of this. I'm sure also that you know this, that there are two different, what would be considered types of diabetes, type 1 and type 2. Type 1 would be less commonly observed. Sometimes this would be called early onset diabetes, because individuals are usually diagnosed with this in their teens to late 20s. This is the result of really an autoimmune disorder resulting in the destruction of beta cells of the pancreas. Beta cells excrete insulin. The immune system destroys those. These individuals stop excreting insulin at some point in time in their life, at which point in time they will start to exhibit these symptoms that I discussed above. And they will then have to be on insulin the rest of their life, okay, from a pharmacological standpoint. Right. Type 2 diabetes, this would be the one that it gets a lot of press now. The incidence of type 2 diabetes has increased within our society over the last couple of decades in really alarming fashion. Just from a description of it, there is a number of really different things that it can be the result of. Certainly it can be the result of something called insulin resistance. Okay? And in this case, the individual has plenty of insulin in the bloodstream, but for some reason, their tissues are not sensitive to insulin signaling any longer. Okay, and that can then lead to the symptoms above, despite the fact that insulin's present. It's just not affecting the tissues in the way that it should be affecting the tissues. Okay, that will lead to what's called hyperinsulinemia, which means that they'll have more than their fair share of insulin, and that can be problematic. Question, yes?
What are some of the ideas or theories that would lead to the resistance?
And we'll talk about those. Yeah, some of those. Okay, so the question is, well, what would be the theories or the ideas behind why a tissue would be resistant? So we'll discuss a little bit of that. We actually, when we went through insulin signaling, I talked about PTEN. I don't know if you remember, but PTEN was a phosphatase that would take the phosphate off of PIP3, making PIP2. It has been suggested that PTEN is overexpressed in some tissue types in a person that's exhibiting type 2 diabetes. So despite the fact that insulin's there, it's signaling, PIP3 is not accumulating and therefore not leading to activation of AKT. And in that case, you can't really get any sustained signaling. Yeah, question?
Is there a difference between excrete and secrete?
Okay, so the question is there a difference between excrete and secrete? No, not in the way I'm using it. Yeah, no. So, okay. All right. All right, so insulin resistance is really one of the hallmarks of this disease. You can see this as well, inadequate insulin secretion or excretion, okay, from the pancreas. In other words, there's something going on with the pancreas that's preventing it from producing or releasing insulin. In this case, though, because it's type 2, the beta cell is still functional. It's not dead. It hasn't been destroyed. It's just not doing what it should in terms of insulin secretion. And then finally, excessive secretion of glucagon or a combination of these things. Okay, the two that I kind of highlighted are the most common, though, seen, okay, that lead to type 2 diabetes. Type 2 diabetes, in terms of causes, certainly there is a very strong correlation with obesity and type 2 diabetes. Okay, 90% of individuals that are a type 2 diabetic are indeed obese, have a body mass index that would be considered obese. So that's certainly an aspect of this. There is a genetic aspect of this as well, though, because there are plenty of individuals that do have a body mass index that would be considered obese that are not diabetic. Okay, so there does seem to be some genetics aspects of it that are not fully understood. Then there would be the other 10% of type 2 diabetics that are not obese at all. Okay, my favorite example of this would be an NFL player, Patrick Peterson. He's actually currently playing as a 6-time Pro Bowler. He plays cornerback for the Cardinals. Yeah, so the Arizona Cardinals. He is obviously in very good shape, but in 2015 was diagnosed with type 2 diabetes. Okay, so quite interestingly, after a really poor year by his standards. Okay, so they had no idea what was going on with him. He was consistently complaining of being fatigued, and finally at the end of that season was diagnosed with type 2 diabetes. So interestingly, he was able to change his diet and has now reversed that disorder. He is no longer considered diabetic based off of these symptoms. And I read an article about him today. I had to look it up again, but this was just like him being interviewed. He said, well, I just ate a lot of fried foods. He said, my wife was from New Orleans, and we eat fried foods all the time, and I just had to change that portion of my diet. So anyway, I thought that was fascinating. So he went on a better diet and reversed that. He lost a lot of weight, too. So anyway, so interesting stuff. So Cause of type 2 diabetes, complicated. Let's say that. Yes?
So I was trying to figure out exactly what you mean by the destruction of beta cells. Is it more like a mutation of them so that they quit functioning, or is it more of a when say like the cells in the pancreas, regenerate themselves?
Yeah. So the question is, yeah. When we go back to this type 1, what exactly do we mean by destruction of the beta cells? Is that a mutation, or are they just not functional? What is that? So the immune system actually attacks them. Okay, and that's what we mean by an autoimmune disease. Right? So the immune system recognizes them as a foreign substance or a foreign cell and attacks them, and then therefore leading to essentially their death. So beta cells then are no longer really. I mean, they're just not there in the pancreas any longer, right? So a treatment of type 1 diabetes, right, certainly you have to just administer insulin. These patients would not be considered candidates for a pancreas implant, okay, or transplant. And the reason is because the immune system will attack it again. Okay, so that's what we mean by destruction. It's really not just mutated, they're just destroyed. Your immune system is your friend, unless it turns on you. Yeah.
Is there a way to distinguish between which cause is causing type 2 diabetes?
Okay, so the question is there a way to distinguish, like between these? Do you mean?
Yes.
Insulin, like inadequate insulin secretion or inadequate or insulin resistance, and the cause of that, or?
Just like, is there a way to determine which?
Yeah, there is. Yeah, we can take blood panels and find out the levels of insulin. In this case, insulin resistance, they actually have higher than expected insulin levels in their bloodstream. Hyperinsulinemia, yeah. So the pancreas is like overcompensating. Like, let's just send out some more. And we'll talk about why that is the case. And whereas this is just not there. So that can be fairly easy to diagnose. What causes that would be more difficult. Other questions? Okay, diabetes. Now let's do this, okay? To understand diabetes, especially type 2, which is really what we're going to be thinking about, type 1 diabetes is not something we'll spend a lot of time thinking about because it's just an absence of insulin. But there's some things about type 2 diabetes that we definitely want to look at throughout the semester. So what you're looking at here is the pancreatic beta cell, and I want to go ahead and discuss then how this cell knows to secrete insulin, okay? And so we're going to start way up here, and we're going to bring forward some information that we talked about that found its way onto the first exam, your favorite transporter, GLUT2 transporter, okay? It was in the liver, and I said it was also in the pancreas. If you remember this there was even an exam question on this, GLUT2 has a really high KT. Okay, kinetically, it's unique in that way. It's got a 17 millimolar KT value. Not only that, but GLUT2 is found expressed in very large quantities in both the pancreatic and the liver membrane. And because of these two properties, these cells will equilibrate the extracellular and intracellular glucose, and they can also respond in fairly rapid fashion to changes in glucose concentration. In terms of the pancreas, that becomes really important because after a meal, of course, glucose concentrations go up. Now, because this is at equilibrium, we could draw it this way, okay? When that goes up here, it will also go up here. And now, the other fashion of this would be this, or not the other fashion, but the other aspect of this would be this hexokinase 4. That's an isoform of hexokinase that has unique kinetic properties as well compared to the other hexokinases. Really large K-half, okay? And because of that, then it's going to respond in a linear fashion. Its rate is going to increase linearly as glucose concentrations go up. So if glucose concentrations double in the bloodstream, they'll double in the pancreatic beta cell, and that will cause then a doubling of hexokinase 4's activity. Right? That will lead to then increased flux through these pathways: glycolysis, citric acid cycle, and oxidative phosphorylation. We have, of course, covered glycolysis. We'll get to the citric acid cycle. We'll get to oxidative phosphorylation later on. End result of those, though, would be this, an increase in ATP production in the cell. So ATP levels will start to rise in the pancreatic beta cell because glucose concentrations are going up in the bloodstream. Now, that ATP, here is where we really want to focus our efforts here. That ATP is going to inhibit. Okay, red X means inhibit in this book. And it's going to inhibit this gene, or this gene product, an ATP-gated potassium channel, so that potassium will no longer go through the membrane. Okay, this would be potassium efflux, meaning potassium is leaving the cell, and ATP will actually stop that, prevent efflux. That will lead to a depolarization, okay, of the membrane that will then activate, put kind of a green circle here, activate a voltage-gated calcium channel, meaning it will open, and that will lead to calcium influx into the cell. That calcium then, and we are not going to go beyond this magical description, okay, will mobilize insulin-containing vesicles that will then bind to the membrane, and insulin will be released. Okay. Obviously, there's something going on in this arrow. And we're not going to look at it. I don't want to bog you down with those things. And that's why I said it's just magical. I mean, that's something that you've probably have recognized about biochemistry is magical. That's what drew you to it in the first place. Okay, this class is even more magical for a number of reasons. Okay, all right, so we'll leave it as a pink arrow, and we'll be happy with that. Calcium increases in the cell will lead to insulin secretion. Okay, all right. So that's how then a beta cell will recognize high levels of glucose and start to excrete insulin. Okay. So go back to type 2 diabetes. If an individual is insulin resistant, the beta cells will secrete insulin, but then there will be no absorption of glucose from the bloodstream because insulin is being ineffective in its signaling. Glucose will remain high. That will lead to greater ATP concentrations in these cells. That will lead to greater insulin secretion, and that will lead to hyperinsulinemia. Okay. Now, let's go ahead and look at this guy a little bit further. ATP-gated channel, or an ATP-gated potassium channel. This is what that would look like to a certain extent. So what I just circled back here is this gene, or this guy here. It's a heterooctamer, meaning it has 8 subunits. It has 4 of these what are called KIR subunits, K-I-R. Okay, so there's 4 times these. Those are responsible for allowing potassium to move through the membrane. There are then 4 SIR subunits. SIR stands for sulfonyl urea receptor. Okay, terribly important, but there's 4 of those as well. Okay. Now off to the right, there's these drugs, glyburide and glipizide. Okay, and I've got a question for you. Let me go ahead and expand this. Okay, so those 2 drugs, and we'll talk about them obviously after this question, glyburide and glipizide, are effective drugs in the treatment of type 2 diabetes mellitus. If I told you that the target of their action is the SIR protein, okay, the SIR subunit of the potassium channel, what do you think would be their mechanism of action? What are those drugs going to do to treat type 2 diabetes, one aspect of type 2 diabetes? Okay, when interacting with a SIR protein. Okay, so what do you think? Text me your answer, what glyburide and glipizide will do once bound to the SIR protein. Okay? All right, I'll give you 2 minutes to discuss that with your neighbor. What do you think? Mark gets that go. Yeah.
Diabetes carries
Yeah, so there's a strong family correlation with it. I mean, it's not one of those things where, like I want to say, I don't think that you could say it's dominant or recessive. There's a complex genetic aspect to it, but there is a family correlation because there are people that exhibit obesity. Like my grandpa was obese for like the majority of his adult life and never had type 2 diabetes. Okay, whereas on my wife's side, anyone of her grandparents that was obese was type 2 diabetic. And many of them died of the complications from that. So on her side of the family, there's a really strong genetic component to it, whereas like on my side of the family, it doesn't seem to be the case. So.
Is that like insulin sensitivity resulting from like a mutation in like the insulin receptor that has been like passed on, or do we not like necessarily?
Well, so we have some ideas, and we'll talk about some of those ideas. We've had some experimental data that would suggest that it's usually not a mutation in the insulin receptor. It's more complex than that. It's typically something downstream of the insulin receptor.
Okay.
Yeah, in that pathway. And some of it might just be simply overexpression. Yeah, 10 seconds left. Yeah, so and we'll talk a little bit about some of those things. Yeah. 2 seconds. Oh, okay. That's it. Okay, so let's go ahead and check this out. All right. Close the channel. All right. Binding to SIR to deactivate it. Okay, inhibit potassium. These drugs help promote the function of ATP potassium channels, decreasing the amount of insulin secreted, decreasing the chances of hyperinsulinemia. Essentially, allosteric inhibition keeps SIR in the same configuration to keep channel open. Okay, so you got open. We got some saying open, some saying close. All right, so let's close the passageway. So let's go ahead and go back to this and look at this thing. Okay, here. If I come back to here, remember. Okay. ATP leads to the inhibition of this. That will close the channel. That will lead to insulin secretion. One aspect of a patient that is type 2 diabetes is that they have inadequate insulin supply. All right. So in that case, if a patient has been diagnosed with type 2 diabetes and they have inadequate insulin supply, they will be treated with glyburide or glipizide that will inhibit this potassium channel. Okay. They will bind to the potassium channel. That will lead to then insulin secretion, and then upon insulin secretion, these individuals, they'll then start to absorb the glucose out of their bloodstream. Yeah?
Do the pancreatic beta cells that are the transporters like having an issue with sensitivity to glucose? Because if glucose leads to glycolysis, ATP production.
Right.
Is what closes the channel anyway.
Yeah, so your question is in regards to, okay, well then, right, is it something... Why is that person have inadequate insulin supply? Is it something up here that's causing problems later on down here that would then allow glipizide and glyburide to be effective treatments of this? And the answer from myself is I don't know. Okay. I don't think, in my readings, I haven't seen anything reported about problems with the GLUT2 transporters, problems with any of these metabolic pathways in and of themselves. There's something else that is causing problems. Okay, so it may be, this is just me, like, just kind of suggesting a possible reason this might be the case. So it may be that there's something wrong with this whole pink magical pathway, okay, that maybe we might even fully understand as a field, where this isn't being screwed up totally, but by giving a little bit more inhibition of this. So ATP is inhibiting this, but then give an additional inhibition by glyburide and glipizide that will lead to greater depolarization, more calcium, and hopefully then will lead to insulin secretion. So that would be a hypothesis. I haven't personally read any of the fact. Okay, so I just don't know. Question?
So I understand the way that you explained it. I guess
Yeah.
my perspective was that I was looking at it from insulin resistance, not from insufficient insulin in the blood. And so
Okay.
I saw it as if you had too much insulin that, not necessarily these drugs, because obviously you just explain what they do, but
Yep.
if there was a different drug that was there to activate it, so then you prevent that, or you keep it, I don't know, it's hyperpolarized or just
Right.
really polarized to prevent insulin secretion, so then you can lower the insulin in the blood which then could possibly reverse the tolerance that's developed. I don't know if that's plausible or not.
Okay, well there is, but it's not through this. It's not a drug that's designed for the pancreatic beta cell. So if you didn't hear what she was saying, well, the way I approach this is that, without having any prior knowledge of what glipizide and glyburide do, isn't it possible then that this would keep this open? So if you think about a patient that comes in that's insulin resistant, right? They have sufficient insulin that we want to reduce the amount of insulin, hyperinsulinemia. Certainly a drug that would keep this open would at least limit the amount of insulin being secreted into the blood. The problem, though, is that wouldn't, cause any more clearance of glucose from the blood. So it wouldn't really treat the disease. It would only treat hyperinsulinemia, but it wouldn't treat elevated glucose levels. Okay, these two drugs don't do that. Okay, but we will talk a little bit about how there's other ways to treat this, and that's my next couple of slides. Yeah. Yeah. Other questions, comments? These glipizide and glyburide, I'm not sure of where the binding site is on the seroproteins. I don't know. I mean, obviously, when this book was published, we didn't have the structure of those, or they would have presented that as well. But they obviously, when you look at them, they look very similar in terms of structure. They're called sulfonylurea drugs. Okay, so that's where this receptor gets its name. Okay, that it's sensitive to those. Now, I will point out, I don't want to spend a lot of time on this, but if you were to look at this on your own time, there are these sites here, and if you have the book, it talks a little bit about these. They have these different sites on these Kerr subunits that are colored in blue. Those have been demonstrated to actually be mutated in a genetic disorder that prevents potassium efflux. Okay, this would be something that's a genetic disorder that if you have a prevention of potassium efflux, then you have hyperinsulinemia in an infant being born. Okay, and the only way to actually, unfortunately, treat that, because it can lead, it's really terrible, because hyperinsulinemia, if everything else is functioning, okay, in an infant that would usually be the case, they don't have insulin resistance. That will lead to hypoglycemia and can lead to a comatose state and lead to the death of the child. All right, so the only really way to treat that genetic disorder when this thing is constantly shut down, okay, because of genetic mutation, is to remove like half of the pancreas. Right? And that individual then has to live without half of their pancreas for really the whole life. And they have this constant insulins being secreted into the bloodstream. There's other mutations that would do the opposite of this, okay? That would be, you see these in red, that prevent ATP from binding. And that will lead to what's called neonatal diabetes. These don't secrete insulin at all. Okay, so there could be a variety of mutations in this. You can't really, those are separate from diabetes. I mean, they're not type 2 diabetes, but there are other genetic disorders that will lead to diabetes-like symptoms. Okay, so. Right, now let's go ahead and talk a little bit about, let's move on, just ask that question. Talk a little bit about other ways to treat this, okay? Glyburide and glipizide are not valid treatments for all type 2 diabetics, because not all type 2 diabetics have, as I've been stating, inadequate insulin supply. They have great insulin supply. Right? They have too much insulin. So how would we treat those individuals? And it turns out that there's a drug called metformin. So let me go ahead and draw you back to this slide. Remember, this slide we talked about the fact that adipose tissues as well as muscle tissue do not have GLUT4 in their membrane, or they have a very small amount of GLUT4 in their membrane in the absence of insulin. Okay, and then insulin signaling comes along in the healthy individual, okay, and mobilizes these GLUT4 transporters and puts the GLUT4, not transporters, but vesicles, and puts the GLUT4 transporters into the membrane. That will lead then to, of course, absorption of glucose from the bloodstream, and glucose levels in the blood will drop in response to insulin signaling. Okay. However, in the individual, and we'll talk about why this might be the case, in the individual that's insulin resistant, we'll talk about how that might be the case later on. But in those cases, how can we get these GLUT4 transporters to the membrane? Okay, and there's actually a drug, and this is our last slide of this lecture. There's actually a drug called metformin. And this is one of the most commonly prescribed pharmaceuticals to treat diabetes currently on the market, metformin. And metformin is this small little drug that will activate in, at least to my knowledge, we don't know exactly how it does this, but it activates AMP kinase, AMP-dependent kinase. And AMP-dependent kinase, as I've mentioned a couple of times, can activate through a different pathway than insulin signaling, but it can activate GLUT4 transport to the membrane and therefore glucose transport into the cells. So metformin will just simply put the GLUT4 from the muscle tissue and the adipose tissue into those membranes, and those tissue types will start to absorb glucose, and glucose levels will start to drop in the individual, even in a fasted state. And that's what metformin does. There has been a demonstration that individuals that are on metformin have a significantly decreased propensity to develop cancer. On these meta-analyses, there's a huge number of patients on metformin, and if you start looking at those populations of people, they're less susceptible to cancer. And that has raised a lot of question in the field if metformin is actually an anti-cancer therapeutic as well. So the jury's still out on that, but interesting. Anyway. All right. Questions on that? So those are 3 possible ways to treat type 2 diabetes. Any questions? Awesome. Let's do this. Let's go to glycogen. Okay, I want to introduce glycogen. Is this it? There we go. So I want to introduce glycogen to you because on Monday I'm going to have you do a worksheet, like I've said, within your groups on glycogen breakdown and synthesis. Okay, so here's what we'll do. I just want to describe what exactly glycogen is, what it looks like, and then we'll just leave it at that, and we'll come back on Wednesday and develop a little bit in terms of the importance of glycogen metabolism. Okay, let me just say this in terms of glycogen metabolism. 2 main tissues have glycogen in them. Okay, the liver, about 10% of the mass of the liver is glycogen, and skeletal muscle, about 2% of the mass of skeletal muscle is glycogen. Okay, the purpose of glycogen in the liver is to regulate blood glucose levels. Purpose of glycogen in the muscle is for instant energy. Okay, the source of glucose for those cells. Okay, now what exactly is glycogen? This is glycogen. Okay, maybe not. It's something like this. Glycogen is a highly branched polymer of glucose. We put this in 2 dimensions. You should really think about this as a 3-dimensional sphere. Okay, and these little branches, each of them represents a polymer of glucose that is about 12 to 14 glucose residues long. Okay. In a glycogen granule, so that's what you're looking at right here, a glycogen granule, there will be greater than 100,000 glucose molecules. So there's a lot of glucose stored in these. Okay, significant supply of glucose. Now, in addition to glucose, a glycogen granule will also have in it protein. Okay, so that's the carbohydrate aspect of this, carbs. But there is a significant amount of protein that would be found in the glycogen granule as well. Okay, and the proteins would certainly be enzymes. Okay, these would be the enzymes that break it down would be found in the glycogen granule, and then the enzymes that synthesize it would be found in this as well. Okay. And then finally, enzymes that regulate, or regulatory enzymes that regulate the breakdown and synthesis. All of those are found within the glycogen granule themselves. They're concentrated in that. Okay. And to be concentrated in that, there is another type of protein that would be considered scaffolding proteins. meaning this. Scaffold proteins are structural proteins that are kind of reaction centers to bring all these other things together. Okay, so we'll talk a little bit about some of these scaffolding proteins that will bring some of the regulatory enzymes as well as the breakdown and synthesis enzymes all into one place so that regulation can occur at fairly rapid levels. Okay. Now, as we look at this, like I've already said, okay, each of these branch, and it's even written on here, represents 12 to 14 glucose residues. Okay. These are attached to each other through what is considered alpha 1 to 4 covalent bonds. I'll define that here in a moment. Each branch point, see this right here, actually let's use a different color, right here, or right here, or right here, or right here. At those branch points, there would be alpha 1 to 6 covalent bonds that allow each of these little polymers to branch off from one another. Okay. Now, interestingly, if we were to look at the structure of starch, it would look really similar. Starch is the storage molecule of glucose for plants. Look very similar, except for the glucose polymers would be a lot longer. Only like 20 to 24 glucose residues. Okay. But most other things, it just, at least from a carbohydrate standpoint, would be the same. Okay. Proteins would be different, and there's not much protein in it. Okay. Now, let's go ahead and look here. What do we mean alpha 1,4, alpha 1,6? Okay. I'm kind of pressed for time. Let's do this, okay? Let's look at this. So you're looking at glucose, okay? And in this is the linear form of glucose. And carbohydrate organic chemistry is coming back to you, right? This is fun stuff, okay? That, what I just pointed at, is the anomeric carbon. And you think to yourself, ah, yes, the anomeric carbon. Thank you for reminding me of the anomeric carbon. What is the anomeric carbon? It's that carbon right there. Okay, so the anomeric carbon is the carbon of the carbonyl of our sugar. Okay, and it can undergo, the anomeric carbon can undergo a nucleophilic attack from a hydroxyl group down the line. Okay, so this can attack that anomeric carbon, all right, and that can lead to one of two possible cyclic rings. In the case of glucose, it's a 6-membered ring because it's the 5th carbon's hydroxyl that attacks the first carbon, the anomeric carbon. Okay? That will lead to either what would be considered an alpha or a beta configuration. Okay? If the hydroxyl group is facing downward, it would be alpha. If it's facing upward, okay, it would be considered beta. Okay. The alpha configuration of glucose is found in glycogen, starch, and amylose. Okay. The beta configuration is found in cellulose, the most abundant molecule on the planet because it comprises most of the cell wall of plants. Okay. You and I can't break down cellulose. We don't have enzymes to do it. Okay. Cattle can. Bacteria can. Right? We can't. Okay. Right. So that's alpha and beta. Now, we said on our last slide in a starch, and not starch, but in glycogen, we have alpha 1 to 4 and we have alpha 1 to 6 linkages. Well, let's go look at this. Okay, so this gets to our glycogen structure a little bit deeper way. Okay, so I'll get to the core here in a little bit. Don't worry about the core. But let's just look at these. Each of these rings represents, it's not a very good representation of the glucose molecules in glycogen. But it's a decent representation, and we'll go ahead and use it. All right, so let me go ahead and do this. I'm going to go ahead and let's focus in on this, and let's number these carbons in this. So this is carbon number 1, 2, 3, 4, 5, and then it's implied here because the oxygen is coming off of it. This is 6. Okay, that would make this 1, 2, 3, 4, 5. It's not drawn here, but it would be the 6 carbons right here. Okay, in fact, it's not drawn anywhere on this. Okay, so the 6 carbon, for whatever reason, they leave it off. But you get the idea. 1, 2, 3, 4, 5, 6. Now, I have just given you then the basis to understand an alpha 1,4 linkage. Okay, it's just an alpha, right? That's there's a linkage between the first and the 4th carbon on adjacent glucose. It doesn't take very much to understand then what is going to be alpha 1,6. And alpha 1,6, if we come up here, this is the 1, 2, 3, 4, 5, right? So we have here 1 and 6. Okay, alpha 1,6 is just the bond between the first and 6th carbon of this. And that's our branch point. Okay. And that's, we could talk about non-reducing ends and reducing ends and all that kind of thing. At the end of these, I guess I should say this, at the end of every branch would be considered the non-reducing end, okay, which is the end with the free hydroxyl group. Okay. And that will be important. The non-reducing end, in terms of terminology, that's where breakdown starts. Okay, is at the non-reducing end. And we build up from a non-reducing end as well. Now, the last thing to say here, are there any questions on that? Glycogen structure? No? Okay, last thing here, glycogen core. So I got these slides out of order. So if you come here, back to this, glycogenin sitting at the core of this. Turns out that to synthesize glycogen, the enzymes that synthesize glycogen need a primer to do that. Okay, and that primer is provided by this, what's called glycogenin. And glycogenin is this. Okay, it's a protein. Hopefully you recognize that as a protein. It's a dimer, a homodimer. And we won't talk too much about it quite yet, okay, but essentially what it does is it will build an eightmer off of it, okay, a polymer that's comprised of 8 glucose molecules, all alpha 1,4 linkages. It's an enzyme, so it will actually catalyze the production of that. But what you would have, it's got this tyrosine here. Okay, don't worry about the chemistry yet. We'll get to the chemistry on the right later on because we haven't really talked about what UDP glucose is. All right, but if we go over to the right, over on the left here, they've got this tyrosine, right? And the active site will actually build from that tyrosine. It will add 1, 2, 3, 4, 5, 6, 7, 8 glucose monomers. Okay, so it'll be this little polymer there, and it will do that because it's a dimer. It's going to do it in both directions. Oops. Okay, all right. And so that's what would be considered the glycogen core. That will be a primer to the synthesis enzymes that you'll be investigating on Monday. Okay. Okay, so have a good weekend. It's supposed to snow on you, so enjoy the snow. Maybe go sledding or something. And I'll see you on Monday. Remember, group work on Monday, okay? So come prepared for that.